Introduction. Biochemistry is a fascinating field that focuses on understanding the molecular mechanisms underlying biological processes. One such process is the biosynthesis and metabolism of histamine, a compound involved in various physiological and pathological reactions throughout the body. In this script, we will explore the intricate details of histidine biosynthesis, histamine receptors, synthesis, storage, release mechanisms, signaling pathways, degradation pathways, as well as clearance, elimination, and the role of histamine in various physiological functions and pathological conditions. We will conclude with a summary of the key points discussed. Let's dive into the world of histamine and uncover its secrets. Histidine, an essential amino acid, serves as the precursor for histamine biosynthesis. Within the human body, histidine can be synthesized through a series of enzymatic reactions. The first step involves the condensation of ATP and PRPP, 5-phosphoribosyl 1-pyrophosphate, by the enzyme PRPP aminotransferase, resulting in the formation of phosphoribosyl ATP. This molecule then undergoes subsequent steps, including the conversion of phosphoribosyl ATP to phosphoribosyl AMP via histidine triad nucleotide binding protein 1, HINT1. The final step involves the removal of the AMP moiety by Hish, leading to the production of histidine. The biosynthesis of histidine is tightly regulated to maintain appropriate levels of this amino acid within the body. Histamine exerts its effects by binding to specific receptors present on target cells. There are 4 known subtypes of histamine receptors: H1, H2, H3, and H4. The H1 receptor is primarily involved in allergic reactions and inflammation, and its activation leads to smooth muscle contraction, vasodilation, and increased vascular permeability. H2 receptors are mainly found in the stomach and play a crucial role in gastric acid secretion. Activation of H2 receptors leads to the stimulation of acid-producing cells, thus increasing the acidity of the stomach. H3 receptors are predominantly located in the central nervous system and regulate the release of neurotransmitters. H4 receptors are involved in immune cell function and mediate various inflammatory responses. Histamine synthesis occurs in several tissues, including enterochromaffin-like cells in the gastric mucosa, mast cells, basophils, and neurons. The key enzyme responsible for histamine synthesis is called histidine decarboxylase, HDC. HDC catalyzes the decarboxylation of histidine to form histamine. Once synthesized, histamine is rapidly stored in vesicles through an active transport process mediated by the vesicular monoamine transporter, VMAT. The storage of histamine ensures its release upon appropriate stimuli. Histamine release can occur through different mechanisms depending on the cell type. Mast cells and basophils predominantly release histamine in response to allergens or IgA cross-linking, leading to degranulation of histamine-containing vesicles. On the other hand, neurons release histamine through calcium-dependent exocytosis upon depolarization. Additionally, certain drugs and substances, such as opioids and radiocontrast media, can induce non-IgA-mediated histamine release from mast cells. Histamine receptors initiate signaling pathways upon binding to histamine. Activation of H1 receptors triggers the phospholipase C, PLC, pathway, leading to the generation of inositol triphosphate, IP3, and diacylglycerol, DAG. IP3 stimulates the release of calcium from intracellular stores, while DAG activates protein kinase C, PKC. H2 receptors, when activated, activate adenylate cyclase through G's protein, leading to an increase in intracellular cyclic AMP, CAMP, levels. H3 and H4 receptors modulate intracellular signaling primarily through G protein-coupled inhibition of adenyl cyclase and modulation of intracellular calcium levels. Histamine degradation primarily occurs through 2 enzymatic pathways, N-methyltransferase, HMT, and diamine oxidase, DAO. HMT catalyzes the methylation of histamine into N-methylhistamine, predominantly in the central nervous system. DAO, mainly present in the intestine, kidney, and placenta, oxidatively deminates histamine, leading to the formation of imidazole acetaldehyde, which is further oxidized to form imidazole acetic acid. These degradation pathways play a crucial role in regulating histamine levels and preventing excessive accumulation. Once histamine is degraded, the resulting products are further metabolized and eventually excreted from the body. N-methylhistamine undergoes oxidative deamination by monoamine oxidase, MAO, to form N-methylimidazole acetic acid. Imidazole acetic acid, resulting from DAO activity, can undergo further metabolism to form 5-aminoimidazole acetic acid. These metabolites are then excreted primarily in urine. The clearance and elimination of histamine metabolites help maintain histamine homeostasis in the body. Histamine intolerance is a condition characterized by impaired histamine metabolism and an increased sensitivity to histamine-rich foods. It results from reduced activity of histamine-degrading enzymes, such as DAO or HMT. The accumulation of histamine can lead to various symptoms, including headaches, flushing, stomach cramps, and allergic-like reactions. The management of histamine intolerance involves avoiding histamine-rich foods and using medications that alleviate symptoms. Histamine plays a crucial role in allergic reactions. When an allergen is encountered, it triggers the release of histamine from mast cells and basophils. Histamine, in turn, causes vasodilation, increased vascular permeability, smooth muscle contraction, and stimulates nerve endings, leading to itchiness and redness. Antihistamines, which block histamine receptors, are commonly used to alleviate the symptoms of allergies. Histamine is a potent stimulator of gastric acid secretion. In the stomach, histamine is released by enterochromaffin-like cells in response to various stimuli, including gastrin. Upon binding to H2 receptors on parietal cells, histamine activates adenylate cyclase, leading to an increase in intracellular CAMP levels. This, in turn, stimulates the secretion of gastric acid, which is important for proper digestion and the activation of digestive enzymes. Histamine has immunomodulatory effects and plays a role in regulating the immune response. It can promote inflammation and attract immune cells to the site of infection or injury. Additionally, histamine interacts with immune cells, such as macrophages and T cells, modulating their functions. Abnormal histamine signaling can contribute to immune-related disorders, such as autoimmune diseases and chronic inflammatory conditions. In the central nervous system, histamine acts as a neurotransmitter, regulating various physiological processes, including sleep-wake cycles, appetite, and cognition. Histamine is predominantly produced by histaminergic neurons in the tuberomammillary nucleus of the hypothalamus. These neurons project throughout the brain, including the cerebral cortex, basal ganglia, and hippocampus. Dysregulation of histamine neurotransmission has been implicated in neurological disorders, such as sleep disorders, schizophrenia, and Alzheimer's disease. In conclusion, histamine, a molecule with diverse functions, plays a critical role in various physiological processes and pathological conditions. Understanding the intricate details of histidine biosynthesis, histamine receptors, synthesis, storage, release mechanisms, signaling pathways, degradation pathways, as well as clearance, elimination, and its involvement in allergies, gastric acid secretion, immune system modulation, and neurotransmission, provides valuable insights into the complex biology of this molecule. Further research and exploration of histamine-related mechanisms may contribute to the development of novel therapeutic approaches for various diseases.